Okay, let's get started. So welcome back to the in-depth session for this afternoon. We already heard some great talks this morning from Kentaro and Christoph. And so in this session, we're going to hear talks from Norman Groves and Joseph McKenna. Before we get started, can I just remind you that if you go onto your Live Now icon on the left hand of your screen, it looks like a little target. If you click on this, and scroll down, it gives you this option of questions. So if you click on there, you should be able to type your question and you should see a little test question as well. It would be really great, especially for our speakers, if you have any questions, to type them in there, and then I'll put them to the speakers, okay? So we're going to make a start with Norman, who is going to tell us, his topic has slightly changed, and Norman's going to tell us all about the targeting of proteins to the inner nuclear envelope in plants.
All right, I assume everyone can hear me just fine. Afternoon for everyone except for me. It's morning for me. So today I'm going to talk to you about research that originated in my dissertation research on nuclear localization signal targeting tail-anchored membrane proteins to the internuclear envelope in plants. So we became interested in this when we were looking at internuclear membrane proteins in animals, of which there are many proteins that have been characterized to interface between the internuclear membrane and the nuclear lamina, as well as chromatin. And these include lamin B receptor, MAN1, emerin, and LEM2. And unfortunately, none of those have homologs in plants, with the exception of LBR, which kind of does, but not really. And we wanted to know, if these proteins do not have homologs in plants, then are there differences in the features which define an internuclear membrane protein in plants? So to start with, we wanted to look back at how proteins are trafficked both on the membrane and outside the membrane to the nucleus. And so in the case of soluble cargo, cargo is actively imported into the nucleus through the nuclear pore complex, through the binding of cargo by caryoferins, alpha and beta, shown here. And these bind nuclear localization signals, which are short peptide sequences rich in arginine and lysine. Once the cargo is bound by importance, it passes through the pore to the nucleus, at which point the importance dissociate from the cargo. So the mechanism by which membrane proteins might reach the internuclear membrane has been theorized. First of all, the pathway by which it gets there is either through peripheral gateways, which are the channels between the nuclear pore complex and the nuclear envelope, where the protein would squeeze between the small channel and then reach its destination, or the central channel by which a protein might span through the assembly of the nuclear pore complex and pass through the central channel and reach the internuclear membrane. There are additionally 4 models for protein trafficking to the INM. These are diffusion retention, INM sorting motif, transport factor mediated, and vesicle mediated. For today, I'm only going to discuss diffusion retention and transport factor mediated. So in the case of diffusion retention, a membrane protein is inserted into the ER, and once there, can freely move between the ER, outer nuclear membrane, and the internuclear membrane. And it'll only be retained at a particular compartment if there's a protein interaction to hold it there. In the case of transport factor mediated transport, a membrane protein is inserted into the ER membrane, but it contains a nuclear localization signal. So that nuclear localization signal is bound by caryoferins. The membrane protein freely moves to the outer nuclear membrane, and then a gating of sorts occurs at the nuclear pore complex. And our cargo containing an NLS passes through presumably the central channel and reaches the INM. And so this has been studied in other systems, but we wanted to know if this pathway existed in plants. So first, we wanted to determine whether this type of pathway exists in plants, and if it did, which we, spoiler alert, found that turns out it does, I wanted to define rules under which it occurs, and then this could potentially be a tool to add new functionalities to the plant internuclear membrane. So to first test this, we used an ER tail-anchored membrane protein that we've worked on previously in the lab, Pickle. And so Pickle has two cold coil domains, a C-terminal transmembrane domain, and a conserved GKNY tail. And to this protein, we affixed the SV40T antigen NLS to the N-terminus. As a control for localization to the outer nuclear membrane, we fused the cache tail for the plant cache protein WIP1. And then we also created a variety of Pickle, which both contained the nuclear localization signal and the cache tail. So for all the data I'll be showing you here today, it'll be transient overexpression assays in the Kochiana benthamiana leaf epidermal cells. And so just to orient you as to what you're seeing, if you go through a cross-section of these cells, these are highly vacuolated. So the majority of the cellular compartments and organelles will be pushed to the periphery. So in a cross-section, you can see the nucleus well, as you can see in the bottom left here. But contents such as the ER, cytoplasm, and actin are just going to appear as fluorescence along the cell periphery. And then if you were to go to the cell cortex, you could see the ER, cytoplasm, or other cellular contents such as actin. Additionally, for everything I'm presenting here today, we quantify this using nuclear localization index. And so here we're just trying to quantify the relative fluorescence on the nucleus compared to other compartments in the cell. And so here I'm drawing line scans on the nucleus, getting the two maximal fluorescence intensities at the nuclear envelope and the two maximal fluorescence intensities in the cytoplasm, and creating a relative ratio here. So we tested each of these Pickle varieties in Kochiana benthamiana. First with Pickle, it behaves like an ER protein. It's localized at the nuclear envelope, but also you see fluorescence along the cell periphery, which are ER contact sites. And if we go to the cell cortex, you can see it's on the ER. And that has a nuclear localization index of about 1.5. When we add our NLS, we get a 3-fold increase in enrichment. We see a comparable enrichment with our cache tail. And these two signals appear to have an additive effect, as NLS Pickle Sit has an increase in nuclear localization index compared to its two compatriots. So next we wanted to test if the yeast protein HED2 could localize efficiently to the nuclear envelope in plants. And the reason for testing this is that a fragment of HED2 containing nuclear localization signals in the transmembrane domain had been shown in yeast to efficiently localize to the nuclear envelope. So we wanted to test this here as well. We saw is that full-length HED2 had an NLI of around 2, which is higher than that of our ER membrane protein. But the linker transmembrane domain fragment, NLS-LTM, had a 2-fold increase in enrichment, which was abolished when you lacked the nuclear localization signals. This is a proof of concept that a previously published fragment could work in plants. So next, we sought to make a chimeric membrane protein that would allow us to test a bunch of different conditions for enrichment at the nuclear envelope. So to do this, we fused the intrinsically disordered linker of HED2 with a transmembrane domain from the plant cache protein WIP1, and then fused an NLS to the N-terminus of that. So our chimeric membrane protein behaves like an ER membrane protein, both in look and in quantification, where it's just below 1.5. Then when we add our NLS, we see about a 3-fold increase in nuclear localization index. Next, we tested if different NLSs had a specialized role for this. So first, we tested 5 different classes of monopartite NLS, these having a single basic cluster of amino acids. It's been previously published that there are 5 different classifications of monopartite NLSs. We tested all of them, and they all were sufficient to enrich our chimeric membrane protein at the nuclear envelope. We also tested 4 bipartite NLSs, including the nucleoplasmin NLS, which has been well studied in the literature. The opaque 2 NLS, this is the NLS from the maize nuclear protein opaque 2, and then the published NLSs from the Arabidopsis sun protein SUN1 and SUN2. And all 4 were sufficient to enrich our protein at the nuclear envelope. So we wanted to use this chimeric protein structure to define rules by which this trafficking may be occurring. So to do this, I already showed you the NLS data, which is sufficient to enrich membrane protein at the nuclear envelope. Then we also tested 2 different linkers between the nuclear localization signal and the transmembrane domain. In the case of the disordered linker, you already saw that data. We also tested a coiled coil domain, and that was also sufficient for enrichment at the nuclear envelope, although we did see an increased number of aggregates on due to the coiled coil domain, and then also it was slightly less efficient in its ability to enrich. Additionally, the NLS, we tested the NLS position relative to the fluorophore. That did not impact enrichment at the nuclear envelope. Then we also tested minimal and maximal distances between the nuclear localization signal and the transmembrane domain. We found we could get down to 50 amino acids without impacting the ability of the protein to enrich at the nuclear envelope. When we got to 10 amino acids, we started to see an increase in nuclear accumulation, but no effect on efficiency of enrichment at the nuclear envelope. We also increased our linker so that we could get above the 60 kilodalton threshold, thought to be the upper limit of what can fit into the peripheral channel of the nuclear pore complex. And this also efficiently made its way to the nuclear envelope. So everything I've told you at this point is nuclear envelope enrichment, and we needed a method by which we could differentiate between the outer and the inner nuclear membrane. And so to do that, we used Airiscan confocal microscopy. And to first test this, we used markers for different cellular compartments. So we used calnexin as our ER protein, and then we co-localized with calnexin either an ONM marker, WIP1, an INM marker, SUN2, or a nuclear peripheral marker, CROWN1. And WIP1 effectively co-localizes with calnexin as we expected. When we co-express SUN2, we start to see a separation in the signals. And when we co-localize CROWN1, we start to see a more significant difference between the 2 fluorophores. We need a way to quantify this. So along with Joseph McKenna, we quantified this data, and to do that, we did line scans through the nucleus, and then we measured the relative correlation coefficient of the fluorescent signals. And so WIP1, SUN2, and CROWN1 each form their own statistical groups. And so WIP1 is the most correlated with our ER marker calnexin, SUN1 less so, and CROWN1 even more less so, showing that we can effectively bin these different cellular compartments based on this correlation coefficient. So we next tested our experimental construct, starting with pickle. And what we found is that our NLS-fused pickle belonged to its own statistical group and was less correlated to the calnexin signal, showing that we can separate and resolve these signals between INM and ONM, and that NLS pickle is accessing the INM. We also did this with our chimeric membrane proteins. We used one monopartite NLS and one bipartite NLS version. And so our no NLS variety is correlated with calnexin, and our monopartite and bipartite NLS both are less correlated with calnexin, indicating they access the INM. And so we have 3 models by which we think our proteins might be reaching the INM. Under one model, our membrane protein would be inserted into the ER membrane, at which point importance would associate with the nuclear localization signal. At that point, this would traffic to the nuclear pore complex, pass through the central channel, and reach the INM, at which point the importance dissociate. The other model the linker between the NLS and the transmembrane domain allows the protein to be more flexible, and it can pass through the peripheral channel, at which point the importance dissociate. A third model, which we can't preclude yet, is that through an unknown mechanism, our membrane proteins are solubly imported into the nucleus and then inserted into the INM membrane. And if such a pathway were to occur, we need a chaperone. Here we've used GET3 as a placeholder, but our data does not preclude this as an option. And so we mentioned earlier in the talk, I was talking about developing a tool to add new functionalities to plant INM. And so a current area of research for us is seeing if we can develop chimeric membrane proteins, which we can artificially reposition gene loci to the nuclear periphery. And to do this, we're testing a system where we could potentially tether an inducer to the INM and then use luciferase as a readout. And so that's something I hope to be able to present to you in the future. But at this time, I'll keep this short. I want to thank everyone. This isn't showing up. Well, my acknowledgements apparently aren't showing up, but thank everyone back at Ohio State. Our research has been funded by National Science Foundation here in the States, as well as the Ohio State Center for Applied Plant Sciences. I work in Iris Meyer's lab as a postdoc now, and the work that I showed you here today was done in collaboration with Oxford Brooks, David Evans, and Kartik Raman, as well as Joseph McKenna, who has since moved to Warwick, and he'll tell you that shortly. And I can take any questions.
Brilliant. Thank you so much, Norman. There. Thank you so much. That was a really fascinating talk. And it's really nice to see it all so neatly summarized. So as I mentioned earlier, if you go as attendees, if you go into your Live Now little target icon and you scroll down, you see a little question icon pop up. If you type your questions in there, and we'll try and answer them. And in the meantime, I can ask a few of my own. Just in actually having that slide up, that's a good one. So you're showing your second model. You're saying that the protein to be imported, the cargo protein, would go through the peripheral channel, but the important complex is meant to associate with the FG repeats. Do we know if are there FG repeats in the peripheral channel? How would that work?
Sure. So the FG repeats are not in the peripheral channel that we're aware of. We have to mention the peripheral channel is an option, although the fact that an NLS makes this more efficient would seem to indicate that this is probably going through the central channel. Now, what it could be doing is the important machinery could make it more efficient in getting to the nuclear pore complex, and then it could pass through the peripheral channel without accessing the central channel. That's something we can't preclude right now. Mm-hmm. We would need to do more in-depth study to really be able to suss out the difference there.
Okay. I had another question. So in some of your earlier images, when you show, for example, where you tested all the different nuclear localization signals and you show enrichment, in some of the images you see these bright dots in the nuclei. Do you generally see deformed nuclear or intranuclear strands, or just these? Can you kind of?
Sure. So we do see aggregates a fair amount of the time. Now, generally, we tend to associate these with using an overexpression system. Now, we don't often see deformities of the nuclear envelope, although it does happen in a minority of cases with particular NLSs. So in particular, the opaque 2 can form some nuclear deformities. I don't have those images in this presentation, but it is, I believe, a supplemental that I included in the paper. So some nuclear localization signals appear to induce deformities more often than others, but it is a rare occurrence relatively.
So is that just because you're pumping loads of protein into the inner nuclear membrane and needs to accommodate it somehow, or?
Well, I mean, it could also be in the case of opaque 2, you're taking a nuclear localization signal from a transcription factor, and that could have unintended consequences. In the case of some of the NLSs I used, these were theoretical NLSs that had been done using modeling and a combination of modeling and experimental data, and so those did not come from an endogenous protein. So it could be that the more NLSs you include from endogenous protein, they could have partners that create these inductions.
Okay. I have one more question.
Yeah, go ahead.
Just quickly check I don't want to take up all the questions, but I can't see any other questions being typed in at the moment. So, okay, one more. So, so right at the beginning, when you had your pickle, so with the pickle sit, you showed that also enriches at the nuclear periphery, but technically, if it's a cache domain, it should sit in the outer nuclear membrane.
Mm-hmm.
So how do you explain that enrichment?
Sure. So, so the enrichment would, in that case, the protein would be following under diffusion retention rules, right? So, normally, pickle would be floating between the ONM and the ER, but now you've added a new protein interaction domain, and so that enrichment can be explained by you're essentially inducing it to interact with SUNS, and so creating an interaction partner creates a place where it needs to be retained, which means that that's going to be the predominant localization now. And the area scan data we show later shows that it's likely on the ONM and not on the INM.
Mm-hmm. Nice. Okay, cool. Right. Then I am going to, are there any more questions? Joe, did you want to ask any questions? I can't see any typed in. Otherwise, we are going to pass the baton to Joe. Thank you very much for this talk. It was really, really exciting. Thank you so much.